All right. Point now?
Yes.
Yeah.
Okay. So what we at the beginning is a review of what we did in part 1. So I just wanted to kind of recap what that is, and this idea that you have dominant and recessive traits, and a lot of the traits we may not actually see unless they are, because they are recessive. So if we have a recessive trait, we're only going to see that trait show up if we have 2 lowercase alleles that are the result. So that's why the square is yellow. But what I wanted to go through here is just kind of a recap of the Punnett square, that if we are crossing 2 heterozygous individuals, so one with a uppercase A, one with a lowercase a, we can designate or kind of do a mock of that cross. And then in the middle is our projected or anticipated ratios or percentages of what the offspring will be. So when you are reporting Punnett square results, you want to report both the genotype and the notype. And by genotype, I mean the letters. So any different letter combinations that you would want to report, like you see down here in the results. And then the phenotype is going to be the characteristic. So whatever the normal trait is would be reported. So maybe it is short hair, or whatever the dominant trait is. Maybe that's a better way to put it. And then that one box that we see, which you could report as 25% as well, would be the recessive trait that we're tracking. So that could be long hair, for example. Animals come in short hair and long hair versions. The individuals that are heterozygous, that don't have that trait that we're looking for, can be called a carrier because they have that lowercase allele, but they don't exhibit that trait. One specific example that we can see is with albinism. So that's a lack of pigment. And you can see that if we do that heterozygous cross, you've got a homozygous dominant phenotype here. You have 50% that are carriers that don't actually show albinism, but they could pass on that gene. And then 25% do show up as having that particular phenotype. And you can report your ratios as percentages. That's usually how I talk about them because we have 4 boxes. Just making sure that you are aware that if I'm asking for the phenotypic percentage or ratio, in this particular example, it's 75% are normal, 25% have albinism. But if I'm asking about the genotype, it would be 25% big A, percent big A, and 25% little a. So there is a difference if I'm asking between the phenotype or the genotype. Any questions on that part so far? No questions so far?
No. Is this PowerPoint going to be posted?
It is posted underneath part 2. Okay. And it's I'm doing of it.
Okay. Thank you.
Yep, no problem. So we're getting into some of the, I guess, different types of genetic inheritance that we can see. The first type is incomplete dominance. And so we can recognize this by the presence of a third phenotype. So instead of just having red and white flowers, for example, that heterozygous individual gives us pink flowers. So think about, I've got red paint and I've got white paint, and if I mix them together, I get pink. So it's neither red nor white, but I get something that's in the middle, and that's how you can determine that you have incomplete dominance. I will tell you that I usually work these problems using the notation that you're seeing here. But what we do see, if you are taking a majors level genetics class, or if you get into microbiology, for example, you may see this particular designation being used, where you have the C and then the uppercase W or R, and then the CRCW would be your pink individual. I won't use that designation in our class. I think it gets a little bit confusing, but I did want to show you that if you see the designation. So we're looking for that F1 generation being all pink. In the F2 generation, so if we cross two pink flowers, we can see both the red and the white reemerge. And so I think it's good, I think it's good practice to go through and do the phenotypic and genotypic ratios on this, like I showed in the other example. So if the question read, what percentage of offspring in the F2 generation were red, what would you come up with?
%.
Yeah, you got it. So 25%. So we've got 25% is red here. We would say 50% is pink, and 25% is white. Any questions on the, on the pink or red or white?
No, I get it.
No. Yeah.
Okay. Okay, cool. The next part is called multiple alleles. So in the previous examples, we just had two choices for the two allele spots in a gene. What multiple alleles is that there are situations where there are more than two options. And our human blood type is a great example of that. There's actually three options that can be combined to give us our blood type. And so it kind of acts as a combination of ways to analyze this. So I would think of the I as kind of the recessive in this case. Only if you have two little I alleles will you have blood type O. So if you have one A allele and one little I, you still have blood type A. And so that would be heterozygous blood type A. This one gets a little confusing because the genotype is very similar to what we call the phenotype because of what we name our blood typing system. With B, you could be big B, or big B O, or I guess the little I here, and that would give you blood type B. And then if you have one of each, then you actually show both of these proteins on the surface of your red blood cells. And so that's how we get our different blood types. And so they say this is codominant, meaning that because I have an A and a B allele, and one is not masked, then I get both of them showing up. I guess an example could be if you had an animal that had spots. So they're not black, they're not only white, they're not gray, but they have black and white spots is a good way to think of dominant. So they call the A and the B carbohydrates. I guess more accurately, it's a carbohydrate and a protein together, and you get a nice visual of the type A on the red blood cell surface, the type B, both of them, and then if you're type O, you have none of them on your surface. And what this gets into is what blood types you can receive if you need a transfusion. So they use a lot of blood typing to do very basic paternity testing. So if the blood typing is pretty cheap and pretty quick, and so if the blood typing possibilities don't match for a potential mother and father pair, then they're not going to go forward and do more expensive, more timely genetic testing. So it's kind of the first level for that. So if we know the father has type B, and it looks like the father is heterozygous because they have one B and one little I here. And if the mother is type A, heterozygous, we've got the A and the I. If we look here in the middle, the offspring could be type O, type A, type B, or type AB. So that doesn't really eliminate anything for us. And you can see down here the results. We can report it as a 1-to-1 to 1-to-1 ratio, or we could do 25% across the board. Either way, on the exam, I try to put both the percentage and the ratio because I know people kind of understand that in a different way, but it means the same thing. Also, when I designate this, I usually put the letter with the B first, and I put the I second, and that just helps remind me that that little I is really recessive. We're not seeing that type O show up. But a good example, I guess, would be if the child was type O, but the Punnett square did not result in type O, then we know the child could not be a result of that parent combination. This slide shows us that for humans, we talked about in part 1 that we have XX and we also have XY for the sex chromosomes, and that's the most often seen results for humans. There are situations where a female could develop with just one X, or we could have XYY, and those are a result of issues with meiosis, and I think we'll talk about that at the end of this unit. But I did want to point out that not all animals and living things have that same system. For chickens and other birds, they have a ZW and a ZZ system, and they have many more chromosomes than we do. For other insects, if it's diploid, it's going to be a female, and if it's haploid, it's going to be a male. So that's a completely different system. And then with grasshoppers, they just have X's. So XX and the female and one X is a male. So I just like to point that out because people generally assume that what we know about humans is true for all organisms across the board, and that's not the case. There's lots of different sex determinations. One other type of cross that we can do, so say the trait is on the X chromosome and not one of the other autosomes, we can actually track that and determine what the percentage of the population or the offspring could have if they are male or if they are female. So this allows us to track traits based on sex. So anytime that is the case, we'll want to automatically put XX for the female and XY for the male. We're tracking hemophilia in this case, so we can see the key. The uppercase H is normal, the lowercase h is hemophilia. And so we can see the hemophilia designated as a superscript. And as we look into the Punnett square, as long as the individual has at least one uppercase H, then they do not have hemophilia. We can see the female carrier down here has one big H and one lowercase h, and that means that she is not a hemophiliac, and hemophilia means that they have uncontrolled bleeding, and they bruise really easy. So she's normal, but she could pass that on to her children. And then what we end up seeing down here is that because the male only has one X chromosome and it's lowercase, then that male is going to have hemophilia. And that's really a great example of why we see a lot more men than women with X-linked recessive disorders. Male pattern baldness is another great example. So they don't have that second X to kind of act as a buffer. Questions about the X-linked Punnett square?
So in
So,
order,
go ahead, sorry.
no, sorry. In order for the thing to be hemophilia, it front, like it has to be the dominant.
No, hemophilia is the lowercase h. So not necessarily in front, but that the lowercase h is the only thing there. So with the men, they only have one X. So if that one X has the lowercase h, then they have hemophilia.
Gotcha.
We don't see one on this example, but for a female to be a hemophiliac, she would have to have two lowercase h's.
Okay.
Oh, okay. Which explains why it's more common for males since they only have one X chromosome.
You got it. Exactly. Exactly.
Okay.
All right. So that example also kind of gets us into looking at pedigrees. And so they track these different traits throughout families. And so females are designated by a circle and males are designated by a square. If the females do not have the trait, then the circle is white. If the female does have the trait, then the circle will be colored in. So in this case, it's yellow, but sometimes they just shade it in. Sometimes they also designate the heterozygous individual, so the carrier, as a half shaded box. So meaning they would draw a line down the center of the circle and just shade in one side. Sometimes they do that, sometimes they don't. So the options that we have here, we'll just track what we see with the upper A. So if we know that one individual is shaded, and so they would be little a, what do you think the genotype of the question mark individual is here? And I can give you a minute to work on that, and other students, if they're watching after the recording, can pause here. So we're trying to determine what the genotype is of the woman with the circle with the question mark. Any thoughts so far?
Is it capital A, lowercase?
That definitely could be a possibility. And why did you choose that one, Brittany?
Because it's not two dominant traits that would make it have a trait.
So you're saying that
I was thinking the same.
Okay.
So it's not two, like, for example, it's not two A's, like two capital A's or two lowercase A's. I'm not capital A's, two lowercase A's, sorry.
Right, so we know that it can't be two lowercase a's because she's not shaded in yellow. I absolutely agree.
Right, but if it was two capital A's, it still wouldn't be with that trait, right? Because it's two dominant.
Yeah, two capital A's would still be colored in white. Okay. And because the sister over here has the trait, we know that the parents have to be heterozygous. So that's kind of step one. So both of those parents are going to be big A, little a. So that means that, like you both, this individual is going to be heterozygous. Less likely, but still possible, she could be big A, and that's because both of the parents are shaded in white. They do not have the trait.
Okay.
And so this is kind of building out, well, what if the impacted sister marries a heterozygous man? And you can see that they can pass on the trait down here, but a majority do not show the trait. And sometimes people know those things ahead of time, especially if it's a disease that is pretty significant, but they also may not know it if it's a characteristic that's really not that important to survival in life, then they're not necessarily concerned with it. Like the male pattern baldness, for example. While that sucks and people probably don't like it, it's not a life or death thing. All right, and then the last little bit of this talks about kind of some terminology that we need to be able to apply, and it talks about pleiotropy. And what this means is that one gene that is making one protein or making one thing happen, sickled red blood cells, for example, can actually have multiple results. So those sickle cells can cause anemia, which then can cause a lot of muscle weakness. It can cause a lot of pain and can cause organ damage as well. So really, one gene having multiple different impacts.
Okay.
We also have this idea of polygenic inheritance, and this is kind of the opposite of what we just talked about. There can be many genes that impact one thing, and those many genes impacting the one characteristic gives us a range of that characteristic. So it's not just red and pink and white flowers, it's red flowers and every shade of red and pink and white in between. And so one great example is human height. We have all different variations of human height throughout the range. We know a majority of people fall in the middle, but we have people who are quite short and people who are quite tall. Another good example of this is skin color. There's multiple different genes that impact the amount of melanin that our body produces. And so you can just see in the human population the very wide range of skin tones that we have, all controlled by genetics and all controlled by melanin. This is a very complex Punnett square. You will not have to do one this complex. We'll just do the monohybrid, but it gives us a good visual of how multiple genes can impact one thing and give us that variation. Not only do genes directly influence our characteristics, but we also can see environmental impact. And so we have some traits that are purely genetically determined, like blood type. We know that other things like skin color can also be impacted by the environment, like sun exposure, for example. If you're out in the sun and you don't wear sunscreen, your skin generally gets darker. And then there are other physical traits that have no genetic basis whatsoever. And a lot of those are culturally or socially impacted. And then the last part here on environmental impact, it can drastically change what the characteristic looks like. So for example, with hydrangeas, if they are grown in basic soil, so we're talking about pH here, then they're more on the pink side. If the hydrangeas are grown in acidic soil, then they're more blue or purple. A lot of hydrangeas that I've seen lately around North Carolina are more white. So that's definitely something that's environmental that drastically changes what that organism looks like. All right. And so there are more slides in the full slide version that's posted in Blackboard. Definitely take a look at those, but those are the ones that I wanted to touch on. I'm going to stop recording.